Good morning, everybody, and welcome back again to the Pineland Speaker Series. Today, we're very fortunate to have with us Kenneth Abel. Ken worked for a long time for Rutgers University down at the Coast Guard Station at the end of Great Bay Boulevard, or also known as Seven Bridges Road. And Ken's going to talk to us today about a book he wrote recently called Beneath the Surface, and is really going to talk about the valley, or the bottom, the floor of the Mullica Valley estuary, where the Mullica River meets Great Bay. And with that, I'm going to turn it over to Ken and learn about the Mullica River Valley.
Thanks, Joel. And thanks for those online for joining us today. Maybe we got more people around because it's a rainy day. But what I want to do is tell you about this book. The book was actually published in August of last year, and in about 300 pages and something like 130 illustrations and images, I try to convey the essence of what we've learned at the Marine Field Station, what we learned about the Mullica Great Bay system, that estuary, over those 30 years. But certainly try to translate it not from the scientific jargon we often use to something that a broad array of people can understand. So this is the cover of the book, and the essence, the study area is the Mullica Valley. So that's what that black outline represents there. This is a vertical map with some exaggeration. We don't have too many mountains in the Mullica Valley, but it does give you some idea of the watershed. And so everything inside the black line drains into the Wading or the Mullica or Great Bay or one of the smaller tributaries. And those outside the line drain into Barnegat Bay, or perhaps Great Egg Harbor River, or even into the Delaware drainage. So we're thinking about this valley, this watershed, and trying to think about it as an ecosystem. It's a coherent ecosystem, and it's related in a lot of ways, and I hope to convey that. And so it's clearer if you look at some of the waterways that are in the system. If you start at the ocean in the lower right, come in through Little Egg Inlet, Great Bay, upstream into the Mullica, but there are a lot of tributaries that contribute to this whole system and drain the whole system, including things like the Wading River and the Bass River, others like Naco Creek and so on. And many of you are probably familiar with some of these, or you may live on one of these. So this is a characterization of the area, of the valley. But one of the things that a lot of people don't recognize is how unique this valley is. So this is a helicopter image with the Garden State Parkway Bridge over the Mullica in the lower center. But if you just look at the whole image, you can clearly see there are extensive marshes. That's the brown or brownish green areas, and the darker areas on the upper horizon are parts of the pinelands. And you don't see in this, besides the Parkway Bridge, you don't see much evidence of humans. And that's part of the reality of this system, and that's what makes it unique. So very, very low population density, and there are relatively few other impacts on the system. And a lot of it is protected by the Forsyth National Wildlife Refuge downstream. As you go upstream, Wharton State Forest, Bass River State Forest, and a whole host of other things that really protect the system. Not much industry. In fact, the most industry that was in the system was a couple hundred years ago, and it's much less now. So really unique, not just by New Jersey standards, by East Coast of the US standards. The common refrain is when I tell people about this system, they say, how can that be? That's in New Jersey. Their mental image is the turnpike and so on. But the reality is there, and we've been studying it long enough. We and others have been studying it long enough to know that it's really quite true. So it's an exceptional system. Getting beneath the surface is difficult sometimes, especially in estuaries, because it's not like being on a coral reef in the tropics somewhere where water clarity is really high and so on. So we use a whole bunch of different kind of approaches to try to understand these systems. So in the upper left is an image from a helicopter. I think we've had about 7 flights over the system in recent years. And that really provides, it's not underwater, but it provides perspective. And I understand the system a lot more just by getting up into the air. And we do that on smaller scales. In the upper right is one of our technicians flying a small drone down at the end of Clark's Landing Road to try to get an idea of the system on a smaller scale. In addition, we do a lot of what we call ground truthing. You know, we do things that help us interpret what we see from the air. So in the lower right, I and a colleague are kayaking up one of the small tributaries to try, like I said, to ground truth, to understand some of the aerial images, and then understand what's at the end of our paddle under the water at different stages of the tide. And then in the lower left shows a picture, an acoustic picture, a side scan picture, and you can actually buy side scan systems, and recreational fishermen are doing it these days, so they can see what the habitats are like. But in that image, all that rough terrain there is an old forest, drowned woody debris from an old forest. And the white line in the center is the area under the boat where it couldn't scan, and then it looks to the right and left. So hence the name side scan sonar. So we're using a variety of approaches to get beneath the surface and understand the whole system. One of the real advantages we have is that being at the old Coast Guard station, old Station 119, since the university took that over in 1972, there's been a lot of research. I myself have been there 30 plus years, and we've used a lot of things in order to sample the system. So there's a listing there. Over 1 million fishes and crabs have been sampled with nets and so on, based on about 49,000 and actually more samples than that now. So we've strained the water in a lot of ways. In addition, we have over 7,000 volunteer hours by people at the station through the Cousteau Reserve that help us understand the system. But one of the most telling things is that picture of the station. So my office is in the lower right-hand corner of that building, and you can see it's elevated. Fish swim under my office on high tide. For someone who spends time studying fishes, that's ideal. You can't get any closer to what's going on. So we have a seat of the pants understanding of what's going on because of that proximity. Scientists would call it ecology of place. Our place is exceptional, and that makes all the difference in the world. But there are lots of other ways of penetrating the surface, trying to understand what's going on. And this is a picture up in Collins Cove in the Mullica. If you look carefully, you can see the Garden State Parkway Bridge over the Mullica when there was some construction going on. But these fishermen are catching mostly white perch, as you can see in the lower left, by hook and line. And a lot of fishermen have a good understanding of the system based on their ability to penetrate the surface with a hook and line. So we learn a lot. We learn a lot from the fishermen in the process. So it's one approach. Another approach is that there are a lot of animals that penetrate the surface, as in this photo here, with, I think my estimate, and you don't see it in the whole screen here, was close to 1,000 birds wading in this marsh pool and around the perimeter. And they do this periodically in the summer when temperatures get really warm and dissolved oxygen goes down in the pools, and the fish that are stressed in there come to the surface, and the birds take advantage of that. But in essence, they're penetrating the surface. So we use their behavior, their activity as a way of understanding how they use the system. And birds do it in a lot of ways. In this photograph by Sue Pooter of a bird skimming, a black skimmer, skimming the surface quite clearly to try and capture mostly fish, both for feeding and involved in courtship behavior and a whole host of things. Other birds that use the system include eagles, and that's more of a consideration now than it used to be decades ago when there were basically none in the watershed. Now there are several nests and nesting pairs that spend time in the system. And we know a little bit about them because we've spent time collecting the remnants of their meals under their nests, not disturbing the birds, but and even we can do it off season when there are no chicks around, but there are a lot of hard parts left over. And that's what you see from some of the prey that they've collected. So in the center of the image, there's some diamondback terrapin shells, and these are surprisingly common. These aren't roadkills; the shells are complete, so they're feeding on diamondback terrapins. In the upper right, under my hand, there are a number of muskrat skulls, and these are also common. Occasionally, you'll get a whole carcass, but a lot of time it's just the skull that you see underneath it. And in the center and lower parts are parts of the skulls of catfishes. So it varies where the nest is and so on. Obviously, more catfishes are being fed on by eagles up in the watershed, but the skulls are distinctive, and you can tell them. So we know that eagles are penetrating the surface, and for a variety of prey, from turtles to mammals to fishes. So again, another approach that I tried to incorporate into the book as a way of getting a view under the surface. There are other animals in the system that penetrate the surface. Bottlenose dolphins, an adult here with a recently born young. They're more common offshore, but they're frequently in the lower portion of Great Bay, and I can sometimes see scenes like this from my office. So again, the advantage of where I spend most of my days is central to an understanding of the system. But there are other marine mammals that use the system as well. So these are a bunch of harbor seals that have hauled out on an island near the fish factory in Great Bay. And these can be quite common. We haven't seen many in the last couple of weeks because they're very seasonal. They show up in general around November. They're here through the winter. And this is a wintertime shot. You can see by the brown grasses. And then around April, they start to leave and migrate back north for the summer. But they're, as you can see, they're a real common component of the system, have been for 15, 20 years, based on work by people at Stockton University, as well as our own. And we've even carried it a little bit further by looking at their scat, their fecal material that they leave on the island there. And we can tell the different kinds of fish bones that are in their scat and help us understand what they're feeding on and where they're feeding on. So they're penetrating the surface and we're looking over their shoulder. I guess that's the correct term. We're keeping an eye on them to understand how they use the system. The other thing we've done a lot is to track fishes and crabs, like the horseshoe crab in the lower left. But these are some of the examples that we've inserted acoustic tags into the body cavity, like for the bluefish up in the upper left, or where a fish has a small body cavity, like the fluke or summer flounder in the upper right. We've actually sewn the tag, which you can see clearly, into the musculature, and that works well. So these tags, they're about the size of a lipstick case, and that allows for about a 2-year battery life. So we've been able to track some of these animals for as long as 2 years. And that has been incredibly insightful in understanding their behavior, both locally and when they migrate. So the other thing in the center left is a river herring, an alewife, which use the system. Center right is a smooth dogfish or sand shark, where the tag has already been inserted. And you can see we got good at this, actually. We could, as soon as we brought a fish on board, we can open it up, insert the tag, suture it back up, and release it. And we were able to do that in 7 or 8 minutes. So these animals are really in good shape when we release them. And we know because we've tracked them moving around for, like I said, up to 2 years. Horseshoe crab lower left, winter flounder in the lower right. So these have provided incredible insights that we couldn't have gotten any other way. And as a further understanding of this, we would track them from boats, note their location. But the other thing we did is establish basically an underwater easy pass system. So listening devices at set places in Great Bay, up in the Mullica. And once these fish, and these tags are individual tags, so not only do we know what species it is, we know which individual is passing by through this easy pass system, just like you driving the parkway. That's how they bill you. They know which vehicle and what individual drove through that easy pass system. So we use the same kind of approach, and it's really been helpful. So for example, here's some data for summer flounder or fluke. This is an image of Great Bay and the Mullica. The Rutgers University Marine Field Station, Rumpus, is labeled there, and we're just inside the inlet. And this is from mobile tracking locations in 2003 and 2004, in the heyday of when we were tracking them. And the shading, the degree of darkness there gives you some idea of the depth. So the darkest is the deepest. In this instance, it's about 15 meters, or sometimes 45 feet or so. And it's shallower in the center part of Great Bay. Whoops. It's shallower in the center part of Great Bay than it is downstream, but then it gets deep again up in the Mullica. So if you just stand back and look at this, you can see there aren't many summer flounder upstream. We weren't able to track them there. And in fact, the only years in which we found them upstream was when there wasn't much rainfall, so salinities were higher and they could go further upstream. By and large, most of the individuals we could detect are in the lower part of the system and in the deeper waters. But that's not the whole story, of course. And so when we use that same sort of easy pass approach in Willett Creek, which is the small creek immediately adjacent to the station, we could track them individually. So what we plotted here, I'll take you through this. This is distance upstream. So this is, again, meters. So 1,000 meters is about 3,000 feet. So it's a small stream. And this is time in days. And each of these tick marks represents an individual day. And this is day 240 in the year. So in the middle of the summer. So the dotted lines show change. This is a tide gauge readout, show changes in changes in water depth or tide height each day. And as you know, in the system, we typically have 2 highs and 2 lows every day. One of each in the daytime, one at night, although it changes. So again, 2 highs and 2 lows, and you see the tides changing very, very regularly. This is the track of an individual fish with a tag. And this is the day it was tagged. We captured in the creek, tagged it and released it. And then it didn't do much the first couple of days. It made a short movement upstream. But on this day, it made a long movement upstream up to about between 600 and 700 meters, and then quickly came back down. And then you see that pattern repeated a couple of times and later as well. But the interesting thing is it's associated with the tides. Now, a fisherman, you may not be surprised by that, but it's nice to see the data. So they're going upstream. They're going upstream on the flooding tide, and they're coming downstream on the ebbing tide. But they did it this time, this flood tide, but not this time and the next one. The reason is this is a night flood tide. Here's a day flood tide. Here's another night flood tide, and the same pattern. So the pattern we discern is that these individuals are coming upstream on night flood tides, going downstream on the ebb, but only during the night, not during the day. And then in a separate study, we captured some individuals, untagged individuals, looked at their stomach contents, and certainly the pattern was clear. When they're going upstream, their stomachs are empty. When they're coming downstream, their stomachs are full. So these fish are using these small creeks in a very specific kind of way. And we couldn't, you can't get in the water and observe them with a snorkel. You have to do something like this. But because of some of the approaches we've learned some of the behavior that is really insightful. We've done some of the same stuff with striped bass. This is Tom Grotus in these pictures. He has been heavily involved in all this. And a lot of the striped bass work has been really, really helpful. And one of the things is that one of the quick and dirty findings is that they tend to stay in the same place for long periods of time. So this is the distribution by depth in the same system, Great Bay Mallika, the Lake Inlet here. The deeper blue colors are the deeper water. So the station is here, and there's about 40 feet of water right here. But by and large, if you look at these red dots is where we've tracked them, where we found them. There's lots of stripers right off the edge of the Sheepshead Meadows here, some up in the channels going up into Barnegat Bay. Not much going on in Great Bay, but then when you get up into the Mullica again, there are lots of individuals detected. And we try to tag fish throughout the system. So it's not a bias of where we tagged them. This we think is a real pattern. So they're highly localized. They prefer deeper water They can deal with different salinities, like in the lower salinities up in the river. But the most intriguing thing, all of it's intriguing to me, but the most intriguing thing is that some colleagues in Maine, working in the Saco River, Maine, southern Maine, set up the same kind of listening array and used the same approach. They tagged striped bass up there. We tagged them in our system. Some of our tagged fish left the system, and we knew that because they passed the sensors going out the inlet, and they would show up weeks to months later in Maine. Some of their fish came down and entered our system. Again, we have individual tags, so we know exactly who it is, where they were tagged, how long they took to make the trip. The incredible thing to me is some of the fish in our system, Great Bay Mallika, left the system, went to Maine, then came back to our system. And when they came back to the exact place where they were as much as 6 months to a year earlier. Now that blows my mind. I would never have predicted that in 1,000 years. But that's what these kinds of tagging programs can show you. So again, we're really penetrating the surface, we're peeling back the surface, looking underwater, but not visually, just with a different kinds of techniques. We also spend time looking at animals in real skinny, that is shallow water, like in this marsh pool with a mummy chog or a minnow. This is the same one that everybody uses for bait for a whole host of things. So we're interested in those systems as well. And when you think about it, things like Great Bay Mallika, there's lots and lots of skinny water there. And it's very, very productive, as you can see by this widgeon grass in the background. And it's an important habitat. So we're trying to cover the spectrum, the range of habitats. But again, the reason we can do this to a large degree is because our location. So the inset in the upper right shows where the Rutgers University Marine Field Station is. And this is a helicopter image that gives you an idea of our location, but also of the kinds of undisturbed marshes that occur in this area. This is critical. Marshes on the East Coast, especially the northeastern coast of the US, have been altered in lots of ways, sometimes paved over, sometimes mosquito ditched, a whole host of things. But there are a lot of marshes in the vicinity of the station that are unaltered. And so again, an exceptional insight. The possibilities are great. The other, well, maybe this is a good time. I told Joel that I would stop and allow people to ask questions, so they don't forget them by the end of the talk. So if there are any questions that I could answer, I'd be glad to do that now. So would this happen live or via?
Yes. Yep. Yeah, if you have questions, please call the number on the screen, and we'll be able to take your questions live on the air.
And if you're still composing your question, we'll answer questions at the end as well.
Okay. Yep. There's the number. Yeah, Ken, that's really neat. You know, I've been fortunate. You know, I grew up in Tuckerton, and I've spent the better part of my life down in that neck of the woods. And I just noticed I saw a lot of my good fishing spots in those two maps you showed, with the deep spots and corresponding to the stripers and the flounder. So that's pretty neat to see. It's great to spend a lot of time in an area and look at it from all these different perspectives. And I really appreciate this program for that reason.
The other side of that coin is when I told fishermen, and we had a lot of help with fishermen because they're better at catching these fish than we are. When I told them what the data looked like and where these fish were, they'd say, I was there last week. I didn't catch anything.
Yeah.
That's why they call it fishing and not catching. It doesn't fish aren't there. And in fact, we can confirm that they are there. So again, another view.
Yeah, and it's neat because depending on the tide and where they're going, they may be there, but like you said, they've probably already fed, so now they're not hungry. So
yeah,
they're just swimming by your lure, and it doesn't look that appetizing to them because their belly's probably already full.
Exactly.
Yeah, I've been very fortunate, in my career and in life, just to see a lot of the country. And I always kind of get drawn back to that neck of the woods down there, Great Bay Boulevard, Seven Bridges Road, and those little creeks that cut across from Tuckerton Bay to Great Bay, or Barnegat Bay to Great Bay. Some of my favorite places in the entire world.
Yeah, no, it's really an exceptional place, no doubt about it. Which is why I'm also considering another book that would focus right on Great Bay Boulevard, Seven Bridges Road, dealing with the history and the ecology. A lot of people have touched on that before, but having basically lived there close to 24/7 for 30 plus years, I think I have some other insights. Yep. Joel, if there are no questions, we could just move on and.
Okay. Yep. We'll just wait maybe another 30 seconds because is a 30-second delay.
Okay.
But yep. I did notice there was around 20 people watching live, so there are some folks out there tuning in.
Good.
All right. Well, I'm going to drop the number. Write it down if you want, or just tune back in with your questions later on, and we'll be able to take them live on the air. Okay.
Thanks, Joel. So the other focus of our research is really on trying to understand the habitats, and the tagging tracking really does a good job at that. But I just wanted to open your eyes a little bit to some of the other habitat specific things that go on. So one of the reasons that we have really such a poor understanding of fishes is because they're underwater. We can't see them. But the other thing is a lot of them bury. A lot of them bury in the substrate. So this is a cusk eel. It's very common in the system. They don't get big. They get maybe a foot long. They have these white barbels under the chin. So they're quite distinctive. But I'll bet you that nobody in this audience has ever seen one before. There are two reasons for that. One, they're only active at night. And two, during the day, they're burrowed in the bottom. So this is an illustration of a cusk eel that's backing into the sand. And they can do this. And they can do this by wiggling their body in this S shape and simply moving the sand around and disappearing under it. And we brought them into the laboratory and watched them do that and so on. So when they're done, there's nothing evident at the surface. So this is common to a lot of fishes. The flounders, a lot of the flounders do it. Summer, winter flounder do it. So it's not an uncommon behavior in fishes. So one reason we know less about them. Of course, blue crabs do that. Other crabs do as well. Lady crabs and so on. So when you're thinking about where these things live, you have to keep that in mind. The same is true for juvenile horseshoe crabs like the ones in this photograph. These are small ones, and I'll venture that most people listening in haven't seen these. You might see their molts. That's how they shed, like the 17-year locusts that are about to invade the area. But they spend a lot of their time under the substrate. They can feed there. They can hide from predators there. So you have to look hard to find these kinds of individuals. And they're buried. The other kinds of habitats that we don't think much about, people think about marshes and think about channels and that kind of thing, but there's lots of variation in the relief you find in these waters. So these aerial photographs, the top one, this is might be Little Sheepshead Creek, just near the station there. But what happens in a lot of these edges, you see this convoluted edge right here. It's not a straight edge often. On one side, the deeper side, you'll have this, not so much on the shallow side. And if you look more closely at this, you see they're calving off like glaciers. So these pieces of marsh grass and the associated peat, the roots underneath, are calving off. They eventually fall into the water, and eventually they keep the vegetation for a while, the grass for a while. But if they stay underwater completely, then the vegetation goes away. So we refer to these as peat reefs, and there's a whole bunch of fishes and crabs and shrimp that associate with these peat reef habitats. So a common habitat, but not really understood by a lot of people. And this is a side scan sonar image of peat reefs on the bottom. So this happens to be a mirror image. I lost my, oh, there it is. This happens, so this piece of peat here is the same as the one here because it's scanning to both sides and seeing a duplicate of that image. But some of these peat reefs, 6 to 8 feet long, 2 to 3 feet high. So they're large structures on the bottom. And given that a lot of animals use this kind of hidden habitat. Another kind of habitat that you see in estuaries, estuaries again are places where salt water from the ocean mixes with fresh water coming from downstream. And there's a gradient, fresh here, little salty, saltier, 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 ocean salinity down here. But in many of these places, the interface between the fresh and salt water is particularly important. And a lot of animals accumulate there, and a lot of animals feed there. So this is a common phenomenon of many estuaries, and it's reflected at the surface sometime, but it's reflected throughout the water column. And then when you think about it, there are a lot of what we say are anadromous fishes like river herrings, like striped bass, that travel along that gradient and go up into the system to feed, to reproduce. And so hopefully if this video works, whoops.
There you go.
So this is a bunch of river herring just below the Route 542 bridge coming out of Batstow Lake. So this is the Batstow River. And this is spring of the year. It's actually right about now. That image was taken right about this time. And those fish are 10, 12, 13 inches. And they've been up spawning up in this freshwater part of the system. And if you go to Batstow on the right day, you can actually see them spawning below the dam. But again, this is another way in which fish use the habitat. But they're spawning up there, and I was afraid they were going to be spawning on my boot because that's how close I was to them. And you'll see spawning activity. So here's another underwater images. And there'll be hundreds to maybe thousands of individuals in the area. And they'll be there a couple of days and then they're gone. And then a week later, 2 weeks later, another group, we think, might show up again. So the males are following the females around, often with their snout at the vent of the female. And if you focus on these, and then here's a group, that was a spawning event right there. They're releasing eggs and sperm into the water, and they're getting ready for another spawning event. There was one there. And another one right there. So again, fortunately, they're spawning in a couple of inches of water, and it's easy for us to film this in the clear waters up in the pine barrens, make it possible to see what's going on. But again, this is just an example of the diversity of habitats that are in the system and diversity of habitats that try to cover in the book. Now I'm going to change gears a little bit and deal with a current issue. Well, the issue has been happening for a while, but it's certainly becoming more apparent and getting people's attention, and that is a changing climate, and in particular, sea level rise. So by and large, we've been sampling water temperatures at the station since 1976. Back then, we only did it daily. Now there are Cousteau reserve data loggers in the system that are measuring temperature, salinity, dissolved oxygen, turbidity, that's the cloudiness of the water, and pH, how acidic it is. And they're measuring it every 15 minutes. So we're getting a much better feeling for how much variability is in the system. But if we talk about climate and sea level rise, we need to talk about a longer term. So in this graph here, this is sea level. It's in centimeters on this axis. And this is from about 1900 to up to the present day. And this is a record of a tide gauge that has been consistently deployed at Atlantic City since 1912, and it's up to the present. So if you look at the change in tide height, every once in a while I lose my arrow, the change in tide height on this axis at Atlantic City over time, 1912 on up to the present, some variability from year to year, but there's no doubt about the trend. Sea level is getting higher. So this is Atlantic City, which is equivalent to it's going to be similar in Great Bay Mallika system. So this is a really good index. But this green line is the global average. So everywhere else in the world. And if you'll note here, the global average about now is around an increase of about 0.5 feet. But in Atlantic City, it's about 1.5 feet. So in other words, 3 times the increase in sea level over this currently relative to the global average. So that's really striking. And that's why studying climate change and sea level rise in our area is really critical, because in essence, we're ahead of the world in many respects. The reason for the difference between the global average and the Atlantic City average is what's known as subsidence. When the glaciers came down thousands of years ago, created Long Island and so on in their process, and there was 1 to 2 miles of ice up there, it pressed the land down up there and caused it to rise further south, i.e. South Jersey, for example. But the glaciers are gone. So that land is rebounding, and the land here is sinking, is subsiding. So we have 2 issues. We have the rising sea level, but we have subsidence as well. So this is why sea level rise, climate change and sea level rise are critical. Climate change is critical because the glaciers are melting again, and that's enhancing sea level rise. And we can see it expressed in a lot of ways, and it helps to get up in the air for this. So this is up near Port Republic, up in the Mullica. I think this is called Otter Pond in the foreground here. And this is lots of marsh around it, but then pine barrens up here. But at the fringe, you see a lot of dead trees. These standing dead trees, often Atlantic white cedar, but oaks and pines as well, are these standing dead trees we and other people refer to as ghost forests, because that's what they look like. But they're common. And if you fly over the area, when we fly over the area, we can see ghost forests are quite common. That's happening because as sea level is rising, salt water is getting further and further upstream, and things like Atlantic white cedar can only grow in freshwater bogs. As soon as they get salt water around their roots and so on, it only takes a couple tides to kill them. So this is why you see so much ghost forest. And here's another image. This is the road. This is the road from the Egg Harbor City area toward Lower Bank. Here's the Mullica. And this area, this is green Atlantic white cedar, living Atlantic white cedar, more here. But this is a vast area of dead Atlantic white cedar. And it really jumps out at you. And this is left over from Sandy. So when Sandy came through, it pushed lots of salt water further upstream. It flooded over these dikes here up into this system, flooded the Atlantic white cedar for a couple of days, and was of sufficient salinity and duration to kill that. And you can drive that road today and those dead cedars are still there. That forest has not grown back. So again, this kind of ghost forest is another indication of sea level rise in the system. But that has been going on for a longer period of time. One of the striking things, and one of the reasons we pay so much attention to Atlantic white cedar, is it's really tough wood. It's really resilient. That's why the area was so important hundreds of years ago in shipbuilding, but it was also used in building houses and so on. It's tough, resilient, but it's light wood, so you can make roofs out of it and not worry too much about putting too much stress on the other timbers. But a lot of these old Atlantic white cedar forests, as sea level rose over centuries, and it's been doing it, are buried under marshes. But when the increasing sea level disturbs those marshes, moves them away, all these old Atlantic white cedar are exposed. So this is a shot from a kayak up in Job's Run off of the Bass River. Also the system. And this is a common phenomenon up in the Mullica. So again, another indication of sea level rise. We refer to these as cedar cemeteries. They've been buried a long time. Now they're being exposed. And just to give you some idea, this is an Atlantic white cedar that was exposed at low tide in one of the tributaries. That paddle on the kayak there, the white part is 18 inches long. So there's a big cedar, and they're quite common when you get up in these systems at low tide. You have to be careful when you get up there so you don't get caught, because they can blanket the whole bottom and keep you from getting out. And that's why most people don't explore it, because they can't get their motorboats up there. They'll tear up their lower unit in less than 30 seconds. So I just want to give you an overview of a lot of the kinds of research that we're conducting But now I'd like to stop talking and answer any questions. And I'm going to quickly plug in my laptop since I'm losing power. Be right back. So Joel, I'm ready anytime you have any questions or if people have comments. Yeah, I'm not sure if Joel is with us or not. And I'm not sure how easy it is for you to call in, but if you want to do that, I'll try to answer your questions.
Hey,
You there, Joel?
hey, Ken, can you hear me now?
Yeah, I can hear you now. Yeah.
Okay. Yep. I have the number up. If anybody's out there with some questions, please feel free to dial in and we'll be here to answer your questions. You know, that's not surprising. Some of that data you showed with Atlantic City. I think in places like Tuckerton Beach and Mystic Islands, where almost every high tide now they deal with some substantial flooding. And it looks like that's just going to continue to increase as time goes on.
Yeah, and you can clearly see it. We have a USGS tide gauge in our boat basin, and we've been recording tides there since 1992. And the pattern is increasing frequency. And we know the benchmark when the road, Great Bay Boulevard, starts to flood because it's doing it with increasing frequency and very regularly. It's just increasing. So that we didn't.
Hello. Hello. Yep. Ken, we got a question. Hello, okay, you're live on the air.
I have a question.
Sure.
I wanted to ask about the water quality in the river, pollution and so on, and the changes in water volume, if there have been any.
Okay, we'll get to that. And if you want, if you lower the volume or shut the sound, you won't have that feedback in the background.
I'm going to hang up.
Okay, we'll take your question off the air then.
You put it on. Yeah, you'll put the on the Zoom. Okay, thank you. Bye.
Yeah. Yep.
Yeah. So the question was about water quality, and I would say in general, the water quality in the Mullica Valley is excellent. We don't have as many people living in the watershed, so there aren't nutrients running off the lawns and all those kind of septic tanks or whatever. So we don't have that kind of issue, and that's reflected in how much oxygen is in the water. So a lot of other places up in Barnegat Bay, especially northern Barnegat Bay, have occasional problems with algae growing in the water, and they have low dissolved oxygen. We don't have those problems in the Mullica. And I think that reflects the fact that the watershed is well protected. The water volume question, I can't answer. It is influenced, we know, by water moving during the period when cranberry bogs are being harvested and that kind of thing. We don't know if that has any negative impact. And then water volume in general is influenced by precipitation. Some people would say that with the changing climate, we're having more precipitation and maybe more water coming downstream. But I hesitate to comment beyond that.
Okay, we have another question. Hello, you're live on the air with your
Water
question.
volume question.
Yes, my name is Gail Rosencrantz. I live in Manhattan. I just love watching all your videos and information. Do you have any tours in this area by boat or on land? I would love to explore it with a group, with a leader like yourself or Ken Abel.
You can join in, Joel, but
Yep.
locally in the watershed, the Tuckerton Seaport has a
Right.
ferry that runs over to Beach Haven and so on, and in Tuckerton Creek. So that's possible.
What's the name of the organization? I didn't get it.
Tuckerton Seaport.
Okay. Could you spell the first name?
Tuckerton and Seaport,
Thank you very much.
SORT. If you go online, that's one word. And you can
Thank you very much.
your schedule there. The other organization that does a lot of outreach kinds of things is the Jacques Cousteau National Estuarine Research Reserve. And they have all kinds of educational
I'm
opportunities.
sorry, what is the first name there? I'm listening to Estuary Research Reserve. What's the name?
I'm sorry, I couldn't hear that.
Tuckerton Seaport.
The other possibility is at the Rutgers University Marine Field Station, we typically have a open house, invite the public in one of the
Thank you very much.
last.
Keep up the wonderful work you do.
Thank you. It just
God bless you.
finished. Yeah, thank you. We just we have an open house one of the last Saturdays in September. We obviously didn't have it last year because of COVID complications, and it remains to be seen whether that will happen this September again. But there are a number of ways to see and experience the watershed. Do you know of others, Joel?
I can't think of off my head. They were the two main, definitely, the Jacques Cousteau National Estuarine Research Reserve. They've got some great programs they've done over the years. And certainly the Tuckerton Seaport, they have a boat that goes across, like I said, to Beach Haven, and they do some other environmental programs along the bay. So both of them would be great options to get up firsthand and see these areas that Ken's been describing. I think that lady was from Manhattan. It's amazing the reach we have with these virtual programs.
That's it. That's impressive. And in response to that, one of the advantages of working in Great Bay Mallika in this really clean system, we've also been working around Manhattan. And so to have the ability to compare a Manhattan shoreline to a Great Bay shoreline is really helpful in interpreting the kinds of things that are going on. So again, it's the place that we have that is really, really critical, really important.
Yeah, along those lines, I know our science office does a lot of water quality monitoring for pH, dissolved oxygen, and all those upland areas of the watershed that are feeding, the Mullica. We seem pretty consistent. The conditions are very stable. What we would consider good pine lands characteristics. Many places with a pH of like 3 to 5.5, so very low pH, low dissolved oxygen dependent on the situation. And so it looks like the protections are really holding in place. And, that ability of the Mullica watershed to handle things hasn't changed because the inputs are still the same. And that's a great sign to see it on both ends, upstream and downstream, that the water quality is still what we would expect or hope for. All right, Ken. Well, on that note, I think maybe we'll stop the questions. Is there anything you would like to close with?
No, thanks for the invitation. Thanks for the providing the capability to share what's going on in our system. And yeah, look forward to more of it in the future. Take care, everyone.
All right. Thanks a lot for tuning in this week. And next week is the 22nd. We're going to take that day off because on April 24th, we're going to have a virtual Pineland short course program, which is going to be called a Pineland short discussion. And we're going to meet with a few people that have spent a lot of time in the area and get their feelings towards why the Pinelands or Pine Barrens are so unique. And look forward for that. That's going to be on April 24th. And that'll be actually the 32nd annual Pineland short course. And we'll see you in about 2 weeks. So on that note, I'm going to stop the live broadcast.
Take care.